molecular impact of binge drinking alcohol. Hi, my name is Eliyana VanDoren, and this summer I was under the mentorship of in Karen Zunzinski's lab studying binge drinking alcohol. And I'm sure you all have an archetype of what binge drinking means to you in your head, but today we're going to be talking about it by its definition. And binge drinking is a pattern of drinking that gets your blood alcohol concentration to 0.08 or higher in 2 hours. To visualize this, you can think of it as 4 drinks for females and 5 drinks for males. By a drink, I just mean a standard shot, a glass of wine, or a glass of beer. But binge drinking is a huge public health concern. And one of the reasons for this is that among adolescents, alcohol is the most commonly abused drug in the United States. And over 90% of the drinking that adolescents engage in is in the form of binging. It's super concerning because adolescence is a really critical period for brain development, and we don't know the effects that this drinking is having on the brain. Another big concern is that females are much more sensitive to the adverse effects of binge drinking than males. And in recent years, the percentage of females with alcohol use disorders has gone way up. And they're more likely to get liver disease and various types of cancer. And not only do we not understand it, but we don't know how to treat it. But what we do know is that withdrawal from binge drinking in adults leads to an increase in anxiety. And we also know that there's a strong association between alcohol use disorders and anxiety. And these are comorbid disorders, very common, which means that the same disorders, both alcohol use disorder and anxiety, are occurring at the same time in one person. And we don't know which way the causal arrow points. So the long-term goal of my lab is to understand the biological mechanisms responsible for the different response to alcohol in adolescents and females to develop targeted pharmacotherapies. And by pharmacotherapies, I just mean things like drugs and other treatments that can help them. And this summer, I was working on a shorter-term goal, which was to examine sex and age differences in the expression of 2 proteins that are known to mediate addictive behaviors. And you decide whether or not it's ethical to subject human subjects to binge drinking for 14 days straight, but science says no. So we use this on mice. We subject mice to binge drinking alcohol. And they're a good model for humans because they're also mammals. We have similar brain structures, and they voluntarily binge drink just like we do. So studies have been done on this before, on withdrawal-induced anxiety, but they've been limited almost exclusively to males. And if you want to know why, you can talk to me afterwards, and I would love to tell you. But we don't have time. So I'll just tell you what they found is that in immediate withdrawal from alcohol, adolescents were resilient to the effects of alcohol. They did not show withdrawal-induced anxiety. And you can see this is a gradient where red is most severe anxiety and it gets less with the lighter color. So they didn't have anxiety at first, but into later withdrawal, we saw at 30 withdrawal some pretty robust anxiety. So it was incubating into late withdrawal. And they saw the opposite for adult males, where they were not resilient at first, they were super anxious, and then that kind of patterned off into the 30-day with not much anxiety seen. So to test this, we had 2 groups. We had a group of water drinkers and a group of alcohol drinkers. And within each group, we had males, females, adults, and adolescents. And I'm going to run you through the process of how we got these mice to binge drink. So first, we habituated them to their cage for 7 days. And they needed to be habituated because mice are normally nocturnal. So they sleep during the day and they're awake during the night, and that makes researching them hard. So instead of us coming to lab at like 3 a.m., we put them in a dark room where it's dark during the day, and so we can study them when they're most active and most thirsty. Then we subjected them to 14 days of binge drinking. So for 14 days, at the same time every day, the mice were either given bottles of alcohol or bottles of water. Then they had a predetermined withdrawal period. So we were interested in how the severity differs between 1 day and, in our case, 70-day withdrawal. So we had them just sit for either 24 hours or 70 days before they were subject to behavioral testing. Now, I'm going to talk about 2 of the behavioral tests that we ran that measure anxiety-like behavior. We can't actually say that they're measuring anxiety because they're not humans and we don't know what they're feeling. So, the first test that I'm going to talk about is called the marble burying test. So, basically, these are the marbles. They're all laid out in this cage with a bunch of their bedding. And a typical mouse who's not anxious will just kind of like shuffle around the dust, but to an anxious mouse, these marbles are foreign objects and they want them gone. So they try to bury them as fast as they can. So what we were measuring was how long it takes for the mice to start burying the marbles. If they take a long time to start burying them, they're not as anxious as the ones that do it right away. The next test we did is called the forced swim test. So mice are natural swimmers, swimming around in sewers. So we dropped the mice into cups of water. And if they were non-anxious, they kind of float around, but when they're really anxious, they thrash. So after that, we did a biochemical test on their brains to see how the proteins and the chemicals were lining up with our behavioral data. So this is mostly what I was working on this summer. And how we did that was, the part of the brain that we were looking at is called the Nucleus Accumbens. And the Nucleus Accumbens, you can think of it as the brain's switch for addiction. When it's really active, the mouse would be more likely to engage in addictive behaviors, and when it's not so active, not so much. And 2 proteins that are really important for the Nucleus Accumbens to make you more, 2 proteins that are important are the Homer proteins. And I'm going to be talking about Homer 2. So they found that in mice with a high expression of Homer, they're more likely to binge drink. But in mice that didn't have any Homer, they didn't binge drink at all. They were actually repulsed by alcohol. So we measured these proteins by extracting brain tissue, taking out the protein, and separating it on a gel like Ricardo and Irfan talked about. And we know how heavy our protein is, so we know where to look for it, but we needed to see how much of it there was. And to do this, we attached an antibody to it. And you can think of an antibody as a lock that fits a specific, or a key that fits a specific lock. It really likes one certain protein and attaches to it so that it's visible for us to see. And the brighter that these blocks are, the more protein there is. So you can see this would be a lot of the Homer, and this would be not so much Homer. So what we found is that our binge drinking model mirrored human data of binge drinking. So we found that the females drank more than the males, which is also true in humans. And we also found that the adolescents drank more than adults. So this was a valid model of binge drinking that we could use for our data. The next thing we found was that irrespective of sex, adolescent binge drinkers displayed an increase in anxiety and late withdrawal. So what we saw before with just the males, where adolescents were resilient to the early effects of alcohol, but that it incubated into late withdrawal, that was applicable to females too. And this was shown by the marble burying test, where this was anxiety-like behavior and that was typical behavior. They found that, so I'm going to orient you to this graph. This is the time that they waited before burying the marble, and this is the withdrawal period. The blue is for water, and the yellow is for alcohol or for beer, whatever makes it easier. And then females are solid, males are checkered. So as you can see, in 1 day withdrawal, you can see that the alcohol drinkers take less time to bury the marbles than their water drinking counterparts. So they're already more anxious in 1 day withdrawal, but look how much it goes down in 70-day withdrawal. So their anxiety incubated into late withdrawal, irrespective of sex. But something interesting we found was that we didn't see this in the forced swim test. We saw that adolescent males in early withdrawal were thrashing around a lot. And this is kind of a novel result. The previous literature has not found this before, but we also introduced a novel variable into this experiment, and that was the inclusion of females. The other experiments had not had females, and we think that maybe because we were using sometimes the same water for the forced swim test, so females would be in there and they pee and they poop in there, and then we put a male in it. So there's already female hormones in that, and we think that that might have an effect on adolescent male anxiety, but further work will be done to see what that really means. But something we found for the adults was that adult anxiety did not lessen with time. In fact, it incubated with time and even got more robust. So the same test, the marble burying test, where this is just an average of both males and females. So you can see that still the alcohol drinkers take less time than the water drinkers to bury, but that time to start burying gets way less until late withdrawal. So they are not resilient to the effects of alcohol in late withdrawal. And we have some initial homework data, but a lot of work has to be done on this. What we found is that adult binge drinkers on withdrawal day 1 have a high density of Homer 2 in the Nucleus Accumbens. And if you recall, high density of Homer 2 indicates anxiety, like drug-seeking behavior, bigger propensity to binge drink. So we have to look at the data from withdrawal day 70, and we have to look at it from both adolescents, adults, males, and females to see the differences. So some conclusions. We know that the sex difference in alcohol intake, where the females were drinking more than males, did not have an effect on the amount of withdrawal-induced anxiety that we saw. So the amount that they drank was not a big factor. But, and then we also did not see a big sex difference. So we did see that there was a difference in age, where the adolescents had an incubation of negative affect, but that was age-related but not sex-specific. And then also, since we saw that the adults were also experiencing this negative affect in late withdrawal, we think that age of drinking onset may not be a reliable predictor of when you experience this withdrawal-induced anxiety. And then lastly, we know that our model of binge drinking is a valid one because it adheres to the standards set forth by the National Institute of Alcoholism and Alcohol Abuse. I would just like to thank my mentor, who's not here. His name is, because he's so busy, he was taking his qualifying exams for his PhD during the month of July, which is when I was with him. But he still was so available and took the time to help me with my project and to walk me through everything, and I'm so grateful. I'd also like to thank Karen, who's here right now, for letting me be in your awesome lab, and we worked with mice today and I got bit. And I would like to thank Sammy and EUREKA and the whole CSET program. Thank you guys so much for being there for us and giving me this amazing opportunity. And that's it.